I will introduce both of you, so if you agree.
Okay, yeah.
So, and Dr. Ahdi Hadrich is a chemist specialized in the design of innovative and functional bio-based materials with a broad and acute interest in the discovery of new, complex and smart systems made from biopolymers. After holding a PhD in materials chemistry from the Rouen Normandy University, he has been an assistant associate professor in chemistry at Paris Frères University. Since 2020, he joined CELABOR as a research and development project manager in the Department of Material, Textile, Packaging, and Bio-based Application. While Dr. Mahmoud Hamzaoui is a chemist specialized in separation science applied to plant and microbial derived natural substances, with a particular focus on the development of solid support-free liquid-liquid chromatographic techniques. After holding a PhD in pharmacy from the University of Rennes, he participated in Marie Curie Action Project. In CELABOR, he focuses his activities on the development of fractionation and purification processes for the isolation of high added value products for cosmetic, pharmaceutical, and food application. They will present as supercritical carbon dioxide, a green and versatile technology for both bio-additive extraction and new bio-based material functionalization. So thank you, and the floor is yours.
Okay, thank you so much for inviting us to present our activities related to the extraction and fractionation by this green technology. So, I will present you one of the technologies used for the during the ECOFUNCO projects, and here I'm just want to mention that I'm replacing my colleague Jeremy Espanyol, which is not available today to present this presentation. So that's why I'm here. This technology is the supercritical carbon dioxide. As a green and versatile technology for both bio-additive extraction and new bio-based material functionalization. So I will start by giving 2 or 3 words about CELABOR, which is a Belgian technological platform for biomass refining and downstream processing. So CELABOR is a Belgian private scientific and technical center based in the Walloon region in Belgium. We are an SME, and we are offering scientific and technical support to companies involved in all sectors related to the circular economy and the bioeconomy, including agri-food, green processing, packaging, textile, and environment. So we have 4 departments, so the extraction and biomass valorization department, materials and textile, environment, and nutrition. So as I said, so we have a technological pilot platform for dedicated to the biomass refining and downstream processing. This platform is divided into big activities. The first one is related to the green extraction processes and purification of small molecules and secondary metabolites from using classical conventional extraction processes, and also green technologies like supercritical CO2 and subcritical water extraction. And on the other hand, we found also our activities related to the materials, biopolymers production, and also green functionalization processes. Here we talk about the cellulose and chitin nanofiber production and functionalization, and also other kinds of biopolymers and polysaccharides. Concerning our activities, so here, as I said before, so in our biomass valorization platform, so we are developing a processing for green extraction and purification technologies from lab to pilot scale. Usually the materials or the starting materials are very various, so we are able to treat bio-based lignocellulosic resources, plants and woods, softwood, hardwood, etc. And we also producing nanofibers, so including nanofibrillated cellulose, nanofibrillated chitin, etc. We also do extraction and purification of metabolites produced by microorganisms. including algae and microalgae, and we use for that the lab scale and also semi-pilot scales technologies. Recently, we also start to focus on the microbial fermentation products in order to produce building blocks and recover them by extraction and purification. So here, as you can see, we in terms of processes, we cover the whole steps from the conditioning of and the pre-treatment of the starting materials, passing to the extraction of the raw materials using extraction and fractionation technologies, and also we are able to purify and isolate a pure metabolite for different sectors, cosmetic and pharmaceutical also sectors. We also have the capacity to post-treat at lab and pilot scale the obtained fractions and extracts by concentration processes, by heating processing, drying, and so on. After that, we pass to the characterization of the obtained pure compounds and fractions by using analytical equipments like HPLC, MS, GCMS, etc. And also we are qualifying the chemical stability and also the biological activity of the obtained extract. So let's move now to the supercritical carbon dioxide. I will introduce you some properties and the applications of this technique in the field of extraction of metabolites, and also applied to when it's applied to the materials. So what is the supercritical CO2? The supercritical fluids is a state beyond the critical point of specific molecules. It has a behavior between a liquid state and a gas state. In the case of the carbon dioxide, which is the most used solvent, green solvents used for the supercritical fluid extraction, the critical point is starting from 31 degrees, and the minimum to be applied will be 73 bars. So here we are talking about a solvent that is easy and conditions that are easy to be reached, and the solvent is not flammable, is non-toxic, is biocompatible, cost-effective for some cases of productions, and also abundant. We have also the possibility to play on the polarity of this carbon dioxide solvent at supercritical conditions by adding some co-solvent in order to modify the polarity and to get moderately polar compounds such as polyphenols, phenolic acids, and flavonoids. Another important point, which is the upscaling possibilities for this technology, which is I want to mention that this technology is not limited to a pilot scale or analytical scale, but is available at industrial scale, which is already at TRL 9. So here, the characteristic of the supercritical fluids are usually characterized by the low viscosity and high diffusivity, which is similar to the gas properties, and also high density and high solvent power, which is very similar to the liquid. So we are combining two properties, and which is making the carbon dioxide at the supercritical conditions a unique solvent, a green solvent for the extraction. Yeah, here is an example of some applications of the supercritical CO2 used for different sectors, usually concerning the extraction activities and the recovery of high added metabolites. We are using CO2 for the defatting of raw materials before extraction as a pre-treatment techniques, but it is a minor application. This technique is used for the extraction of food ingredients, especially aroma, non-polar dyes like carotenoids, for example, vitamins like tocopherols, and also specific lipids and fatty acids, especially from microalgae like omega-6, omega-9, and omega-3. This extraction using CO2 at supercritical state is also used for remove the organic solvents after the extraction using conventional solvent extraction. It's also use it for the recovery of some interesting aroma from the fermentation, after fermentation, and of some beverages like beer, for example, or other kind of beverages. We also apply this technology to recover and also to deodorize some natural liquids. Here again, I give you an example. Some cosmetic oils, for example, argan oils, and also some oils that are used for cosmetic application are usually characterized with very characteristic odor, and sometimes it's not, those odors are not welcome for the cosmetic formulation, so we need to remove them by applying supercritical CO2, and we will recover directly the deodorized liquids and oil to be directly formulated without any other processing step. Recently,
excuse me, Mahmoud, you
yes.
have 5 minutes left.
Oh yeah, okay, I have to speed. So here some other application also when we use the supercritical CO2 in the materials for example, to impregnate some dyeing and pigment and colorant for textile, for example, and some other fibers. It's also used for the to control the fabrication of biological active nanomaterials. And also here in the case of the ECOFUNCO project is interesting technology to be applied for the coating and exfoliant of the intercalation of lighted materials. So here in terms of the ECOFUNCO application, at CELABOR we have different kinds of equipment. Here we start from the lab scale development, and we can go up to the semi-pilot extractor and using our equipment here at CELABOR. After the logical, let's say, step after the validation at the semi-pilot scale, here using our pilot equipment here at CELABOR, we can find the possibility to go and to the full industrial scale using 1.6 ton extractor vessels that are available here in some industries in Europe. This is an example for the work that we have done on the tomato seeds in order to recover the fatty acids from the tomato seeds here in the frame of the ECOFUNCO project. So here, after this optimization using a design of experiment, we defined it and we found the optimal CO2 extraction parameters, and those extraction parameters were validated at a lab scale. And we found very good recovery and yield of the fatty acid fractions, and we have done the characterization of the obtained fractions, and also we we moved afterwards to the pilot scale upscaling experiment, and here we have found that it's perfectly upscalable, and we found approximately the same yields and also characteristic of the final oil. So I will give the word now to my colleague Ahdi to continue the last 2 slides.
Thank you so much, Mahmoud. Yeah, I will be. I will try to be rapidly to not depass the time. So the extracted fatty acids was further used to functionalize the ketene nanofibers. And this is to improve the barrier properties of the nanofibrillated ketene in presence of humidity, and the compatibility also with hydrophobic substrate made from bioplastics during coating process. So for this purpose, the approach was based on the production of nanofibrillated ketene extracted from mushrooms and shrimps, and this was done by pre-treatment and also mechanical defibrillation of the ketene as the first step, and then its surface modification using again the supercritical CO2 as the reaction medium to substitute the organic solvents, and for that the fatty acids extracted from tomato seeds were used as a hydrophobic molecule. Next slide, please, Mahmoud.
Yes.
Thank you. So the reaction was successfully realized and confirmed by infrared spectroscopy with the appearance of new peak at around 74 reciprocal centimeter, which corresponds to the stretching vibration of carbonyl bond of the link established between the fatty acid and the ketene nanofibers. Another rapid test also that we can see throughout the following video. If you can just go to the video, Mahmoud, please. Yeah, it's a sample drop water test. And here, as we can see on the right, it was the ketene nanofibers which was modified by fatty acid, and we can see that the water was not absorbed by the product, and which confirms the hydrophobic behavior of the ketene nanofibers grafted by fatty acid. However, the product on the left was the NIAT nanofibrillated ketene, and we can see also that the total quantity of water was absorbed by this nanofibrillated ketene, which confirms again the success of the crafting reaction using the supercritical CO2 as a green technology and innovative technology to graft hydrophobic molecules such as fatty acids into nanomaterials. So for future, now we are thinking, since we can extract the fatty acids by the supercritical CO2 and then go to the functionalization of nanomaterials by supercritical CO2 and using this fatty acids, why not now thinking about the creation of one line system, including the extraction, but also the hydrophobic surface functionalization of materials using again the supercritical CO2. So thank you everybody for your attention.
So thank you.
Sorry for the time. I think.
Yeah. We have a question. Can degrafted chitin be coated on paper and film?
Can we graft the
Can degrafted chitin or ketene, sorry for the pronunciation, be coated on paper and films?
Yeah, of course. After the coating of ketene on the films or on the paper, we can also use the supercritical CO2 and the fatty acids to graft them on the substrate pre-coated by the material, the nanofibrillated ketene. Yeah.
Okay.
It's an approach that we can apply using the supercritical CO2.
Okay, thank you so much for your answer. And thank you for presenting the properties and application of supercritical carbon dioxide.